Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for the DBT-JRF because only 9 days are left. So this is the day 24 of the free BET crash course in which I am going to discuss some of the important questions from the genetics portion. So stay tuned, and also I have a request for all of you. Kindly watch the video that I am going to publish it on the 12th of April, because the single video will comprise so many important topics that is going to be asked in this DBT-JRF 2024 examination. So watch that video and hit the like button if you like my efforts, and subscribe my channel if you want more. So let's start with the very first question. So this is the question number 1. So question number 1 says that a cross between two true breeding lines, that is one with dark blue flowers. So let's represent the dark blue flowers with capital B. This is for blue flowers. And one with bright white flowers, let's represent it with small b for white flowers, which produces F1 offspring that are light blue. It means F1 generation offsprings are light blue, that is capital B and small b. Now, when the F1 progenies are selfed as a 1:2:1 ratio of dark blue to light blue to white flowers is observed, what genetic phenomenon is consistent with these results? So, when the F1 offspring are processed to the F2 generation, so a ratio of 1:2:1 was obtained. So, you have to name that which genetic phenomenon is occurring in the said question. So you must be aware that this is not the epistasis, this is the incomplete dominance, this is not the codominance, and this is not the random mating. So correct option is option B, that is incomplete dominance. So in the question it was asked that when true breeding blue flowers are crossed with this white flower that is of homozygous nature, so in the F1 generation all the plants were comprising the light blue colored flowers. So when the F1 is just self crossed, so in the F2 generation a ratio of about 1:2:1 was obtained, indicating that one is dark blue and two flowers are of light blue color and one is of white color. So what does this phenomenon indicate? So you have heard the term incomplete dominance. So in genetics, incomplete dominance is a phenomenon in which the heterozygote, that is this capital B and small b, has a phenotype intermediate between the phenotypes of the homozygote. So it means it will carry the feature of the dominant characteristics as well as the recessive characteristics, and therefore the phenotype of the F1 generation plants will be the blending of the both the parents, that is the recessive parent and the dominant parent. Therefore the mixing of the dark blue color with the white color gives the phenotype of light blue color in the F1 generation, and when it was selfed it gives the ratio of 1:2:1, that is the genotypic ratio and the phenotypic ratio in the incomplete dominance is same. So the correct option is incomplete dominance. Now let's move on to the question 2, that is mutation which occurs in the body cell which do not go on to form gametes can be classified as. So this was the very simple question. The mutation which occurs in the body cell which do not go into the formation of the gametic cell are known as the somatic mutations. So the correct option is somatic mutations. So basically in multicellular organisms, we can distinguish the two broad categories of mutation. That is, mutation can be classified as the germinal mutation or the germline mutation or the somatic mutation. So the germline mutation is a mutation which takes place in the gametic cell, and the somatic mutations arises in somatic tissue which does not produce gametes. So the cell which is not involved in the gametic production, and in that cell, if an mutation occurs, that is known as the somatic mutation. Now question number 3, that is polyploidy refers to. So we have already discussed what exactly is polyploidy, aneuploidy, and also I have shared the different flashcards. So do polyploidy refers to the extra copy of a gene adjacent to each chromosome? An individual with complete extra set of chromosome? Yes, this is the correct option. A chromosome which has replicated but not divided? No. Multiple ribosome? No. An inversion which does not include the centromere? No. So there is an easy way to go to the correct option, that is you have to read, and if there is an extreme option, so you don't have to read it completely, just eliminate it and save your time so that you can scan out of the 150 question. You are familiar with the that question. So like the option number D says that the multiple ribosome. So ribosome का यहाँ कुछ काम ही नहीं है, तो आप ये ऑप्शन को ही कट कर दीजिए. Now an inversion का भी कुछ काम नहीं है polyploidy में. Polyploidy refers to the an individual with complete extra set of chromosomes. So यहाँ पे easily आप आंसर पिक आउट कर सकते हैं अगर आप स्टेटमेंट को बहुत अच्छे से पढ़ेंगे तो. सो करेक्ट ऑप्शन इसका है ऑप्शन बी. So question number 4, which says that which component of transcribed RNA in eukaryotes is present in the initial transcript but is removed before translation occurs. So out of the given option, you have to find out that which of the following component is present in the initial transcript but it is removed before going on to the translation process. यानी कि initial transcript में वो present रहता है but translation होने से पहले उसे eliminate या remove कर दिया जाता है. So is it a intron? Yes, intron ही है वो. 3' poly A tail? No. Ribosome binding site? No. 5' cap? No. Codons coding for the protein to be produced? No. So introns are removed before the translation occurs. Now question number 5, that deals with X chromosome inactivation. So now let's find out that why the X chromosome in mammals is inactivated. So basically we are aware with the fact that the male have the 1X and the Y chromosome. So the males have 1X and 1 Y chromosome and the females have 2 X chromosome. So this X chromosome inactivation, so this 1X chromosome inactivation ensures that the males who have a single X chromosome and the females consisting of the 2X chromosome receive the same functional dosage for the X-linked genes. It means the X chromosome are carrying so many genes on it. So if that both X chromosome will be expressed in the females, there may be that dosage imbalance. It means कि जो females होंगी उनके में जो X-linked chromosomes दोनों ही अगर एक्टिव होंगे तो वो दोनों के ही जो लिंक्ड जीन्स हैं वो भी एक्टिव होंगे. इट मीन्स कि कहीं ना कहीं बहुत ज़्यादा हाइपर जीन्स का फंक्शनैलिटी देखने को मिलेगा फीमेल्स में एज कम्पेयर टू द मेल्स बिकॉज दे हैव ओनली 1X क्रोमोसोम. सो टू मेक अ बैलेंस एंड टू मेक अ बैलेंस of the functional dosage for the X-linked genes, the 1X chromosome of the females are inactivated. Now let's move on to the question number 6, that is QTL analysis is used to. So firstly you should know about the QTL. QTL stands for the quantitative trait loci. So now what is the QTL? So the QTL is the quantitative trait loci and it is the chromosome regions containing genes that influence a quantitative trait are termed the QTLs. It means कि chromosome का वो region जो ऐसे genes को hold करता है जो कि influence करते हैं quantitative traits को, that are termed as the quantitative trait loci. So you have to told that QTL analysis is used to identify RNA polymerase binding site? No. Map genes in bacterial viruses? No. Determine which genes are expressed at a developmental stage? No. Identify chromosome region associated with a complex trait in a genetic cross? Yes. Determine the most rapidly evolving parts of genes? No. So the QTL is used to identify chromosome region associated with a complex trait in a genetic cross. As I mentioned, QTL क्या होता है? QTL क्रोमोसोम का वो रीजन होता है जो कि कंटेन करता है ऐसे जीन्स दैट इन्फ्लुएंसेस अ क्वांटिटेटिव ट्रेट और अ कॉम्प्लेक्स ट्रेट. सो QTL का फंक्शन ही ये होता है कि इट आइडेंटिफाई क्रोमोसोम रीजन एसोसिएटेड विद अ कॉम्प्लेक्स ट्रेट इन अ जेनेटिक क्रॉस. सो करेक्ट ऑप्शन इसका है ऑप्शन डी. सो दिस वॉज ऑल फॉर टुडे. आई होप आपको आज का वीडियो अच्छा लगा हो. सो हिट द लाइक बटन एंड सब्सक्राइब माई चैनल. And again, I'm requesting, please do watch the video that will be published on the 12th of April, because that single video will give you a lot of marks in this upcoming DBT-JRF examination for sure. Thank you so much for watching.